जी स्वाति।
I, Swati Jain, National Joint Secretary, SPSR, on behalf of SPSR, welcome you all to attend a national webinar on the topic Countering the Rise of Superbugs through Antimicrobial Resistance by noteworthy speaker Dr. Geer Mohammad Ishaq sir. Let me extend a hand of gratefulness to highly learned Professor Dr. Shashi Alok, President SPSR, to have given me an opportunity to transact a highly informative webinar on a significant topic today. Welcome, Dr. Shashi. I would also like to welcome our eminent keynote speaker, Dr. Geer Mohammad Ishaq sir, Associate Professor at Department of Pharmaceutical Sciences and Coordinator at Human Resource Development Center at University of Kashmir. SPSR feels highly honored to again have got an opportunity to share a part of your vast knowledge today, sir. I also heartily welcome the entire team of SPSR, Secretary SPSR, Mrs. Monica Sabharwal, National Joint Secretary SPSR, Dr. Abhishek Mathur, who will be moderating today's webinar with me, and all other dignitaries of SPSR who are diligently working to strengthen the foundation of SPSR. Since this webinar is organized by SPSR, I would like to elucidate its purpose. Society of Pharmaceutical Sciences and Research is an educational non-profit organization. It is committed to encourage the study and practice of pharmacy education and research. The mission of SPSR is to promote the pharmaceutical sciences in all aspects. Our vision is to promote and advance the science of pharmacy and its applications in India. We look forward to promoting and facilitating the acquisition, dissemination, and exchange of pharmacy knowledge and information about the practice of pharmacy and pharmaceutical sciences. Now I request Dr. Shashi Alok to kindly take the lead.
Thank you, Swati Jain. A very good morning to all. Myself, Dr. Shashi Alok, President SPSR. On the behalf of Society of Pharmaceutical Sciences and Research, I welcome Honorable Speaker Dr. Geer Mohammad Ishaq sir, Associate Professor, Department of Pharmaceutical Sciences and Coordinator, Human Resource Development Center, University of Kashmir, Jammu and Kashmir. I also welcome to Mrs. Monica Sabharwal, Secretary SPSR, today's webinar moderators, Mrs. Swati Jain, Joint Secretary SPSR, Dr. Abhishek Mathur, Joint Secretary SPSR, Office Bearer of SPSR, and all the virtual national and international participants who are presented here in this SPSR National Webinar. So viewers, SPSR today going to organize a national webinar on countering the rise of superbugs through the antimicrobial resistance. So today we have with very renowned speaker for this enlightening talk. I again welcome to Honorable Speaker Dr. Geer Mohammad Ishaq sir, and thank you sir for accepting our invitation for this national webinar. Now I would like to request Mrs. Swati Jain kindly introduce today's eminent speaker. Over to Mrs. Swati Jain.
Thank you so much, Dr. Shashi. For the ones who have not been fortunate to have heard him before, I would like to read out Sir's profile for them. Dr. Geer Mohammad Ishaq is Associate Professor at the Department of Pharmaceutical Sciences, University of Kashmir, Jammu and Kashmir, India, where he has been working for the last 18 years. He has been instrumental in designing and starting a postgraduate program in pharmacy practice at the University of Kashmir and is presently in charge of this postgraduate program offered by the University of Kashmir. Dr. Geer has also served as a drug control officer with the government of Jammu and Kashmir, India, for a period of about 3 years prior to joining the Kashmir University. He holds a doctorate in pharmacology and has research interests in pharmacovigilance, pharmacy practice, pharmaceutical care, drug utilization evaluation, pharmaceutical policy, quality control, and rational use of medicines. Dr. Geer has so far published 48 research papers, 46 abstracts, and more than 100 popular articles, and presented 42 papers in various national and international conferences. He and his team has won 5 Best Paper Awards in various conferences and journals. He has delivered more than 100 extension lectures on various topics outside his department, besides reviewing numerous papers for various national and international journals of repute. He has reviewed and revised the drug policy in his home state of Jammu and Kashmir. At present, Dr. Geer is also dispensing his additional duties as coordinator, Human Resource Development Center, University of Kashmir, and in this capacity, he has organized 19 teacher training courses as course coordinator. Dr. Geer has also been appointed as member expert committee on antibiotic utilization under National Drug Utilization Evaluation Program of India by the Indian Council of Medical Research ICMR, New Delhi. He has also served as country leader India BRICS Medicines Alliance and as Joint Secretary North Zone Association of Pharmaceutical Teachers of India APTI. Recently, Dr. Geer has been appointed as advisor to the Pharmaceutical Price Monitoring and Regulating Committee of the Drug and Food Control Organization of Jammu and Kashmir. Sir's magnificent personality itself speaks volumes about the expertise he has, and that's the reason why we have got a staggering figure of over 3,100 registrations today, not only from India, but from more than 26 different parts of the world, like Thailand, North Macedonia, Egypt, Romania, Ukraine, Malaysia, Nigeria, Kingdom of Bahrain, Bangladesh, Pakistan, Saudi Arabia, Sri Lanka, Nepal, Azerbaijan, Syria, Philippines, UAE, Macau, Iraq, Yemen, Ethiopia, Yemen, Morocco, Kenya, Mauritius, United States, and others. Now I request Dr. Abhishek Mathur to kindly give us the welcoming notes and give a briefing of today's webinar's topic to our audience before starting today's webinar with the keynote speaker Ishaq sir, having vast knowledge in this area. Over to you, Mathur sir.
Thank you, Swati ma'am. It's a great privilege for SPSR and for all of us to have Dr. Ishaq sir with us on such a great topic, the antimicrobial drug resistance and the overcoming the drug resistance of superbugs by some novel molecules or natural molecules. So how we can overcome this problem, this is actually a question mark, because in today's scenario definitely the antimicrobial resistance or drug resistance is being prevalent and is being spreaded throughout the country, throughout the globe, and these all microbes are found to be drug resistant against some antibiotics, the conventional antibiotics which we are using. The drug resistance is because of the kind of conventional antibiotics, the normal medicines we are taking, the normal antibiotics we are taking in our daily usages without consulting doctors or physicians. And sometimes there are some people, there are some lobbies also who are taking the medicines or drugs for each and every thing, and they are not taking the proper advices from the doctor, and they used to take the medicines and drugs. So coming to drug resistance is being spreading, and there are several microbes like Staphylococcus aureus, Pseudomonas aeruginosa, and there are different other strains also which are found to be drug resistant against vancomycin, the methicillin resistant as aureus strains are there. There are Pseudomonas aeruginosa which are found to be drug resistant strains. So antimicrobial resistance is threatening the effective prevention and treatment of ever increasing range of infections which are caused by any bacteria, parasites, viruses, and fungi. Now, today's scenario, as we are suffering from the COVID, we are having the vaccine. But anyhow, we are also aware about the information that this strain is having the mutation, is causing the mutation, is itself mutated, and different types of mutated strains are been spreading throughout the globe. So when one person is taking the vaccine or suppose any drug, so there is also a question mark that how much aspect this particular drug or vaccine will affect this particular strain from which the person is affected. So this antimicrobial resistance is not restricted towards the bacteria. It is restricted towards the viruses. It is restricted towards the fungi. It's restricted towards the parasites. So all antibiotics, antivirals, antifungals, antiparasitics, and these are basically categorized under the category known as antimicrobials, and they are meant for the prevention and treatment of infections in humans and animals, and different microbes, different other plants, etc. So because in agriculture also we have found we are getting different other problems, and which the specific pathogen is found to be drug resistant. So there is a need to identify and explore different natural molecules, and to identify some compounds, some natural substances available in the nature, which might be found to be antimicrobial or antiviral or anti parasitic, and there is a need to study some molecules like that, because if these conventional antibiotics will not work further, there is an urgent need to identify such molecules which can cause the proper treatment and proper remedial action. So coming to this particular topic, and we are having Dr. Ishaq sir with us. So over to Ishaq sir for the lecture. Thank you so much, sir. Thank you, SPSR.
Thank you very much, everyone. Dr. Shashi Alok, President, Society for Pharmaceutical Sciences and Research. Dr. Monica Sabarwal, Secretary, Society for Pharmaceutical Sciences and Research. Dr. Swati Jain, National Joint Secretary, SPSR. Dr. Abhishek Mathur, National Joint Secretary, SPSR. I'm wholeheartedly thankful to the entire team of SPSR for once again giving me an opportunity to talk about a very important topic. Earlier, I was here on this forum talking about the NDMA impurities in medicines. And this is the second talk which I'll be delivering today on the rise of superbugs, which is a crucial issue, a global public health challenge. In fact, the World Health Organization has listed the antimicrobial resistance among top 10 global public health threats that the humanity is facing. And there are estimates that at present there may be around 7 to 8 lakh people dying every year as a result of antimicrobial resistance. But Jim O'Neill of UK had predicted in 2016 that by the year 2050, there may be 10 million deaths as a result of antimicrobial resistance. And it may cost the world economy a whopping $100 trillion. And not only that, it affects our development, our progress, our environment. A whole lot of issues are interconnected, which will be drastically affected. Therefore, this is the right time to discuss and deliberate on this very important topic, and not only to create awareness and know the basics, but also to search and explore the ways and modalities by which we can control this menace, by which we can prevent it, by which we can curb it, and to evolve novel strategies, innovative approaches by which the antimicrobial resistance could be curbed. So as some of the novel approaches have been listed by Dr. Abhishek Mathur, that is exploring the new entities from the natural world, from natural products. So dear participants, before I start my presentation, I would like to tell you that many people view this antimicrobial resistance as part of evolution and say that this is bound to happen because every organism on the face of this earth attempts to survive and does whatever it can to survive. So this antimicrobial resistance is nothing but the survival pressure, selection pressure, because microbes also want to survive, and given the population density at a particular environment, they try to mutate, they try to adopt genetic material, they try to evolve and get transformed into new forms. They try to make newer components, newer proteins, which may not be detected by the antibiotics. And thereby they try to acquire this resistance. So we'll see what is antimicrobial resistance and how it can be tackled, because it might seem a very small issue to some of the people who think that we just take a few tablets of antibiotics or antimicrobials, and that may not sum up to a big amount, big quantity. But the figures tell us that annually 0.1 to 0.2 million tons of antibiotics are used worldwide, which is a very huge quantity. And this is affecting the health, the resistance of microbials immensely. So these are the issues which we'll be talking about from an evolutionary perspective, from the drug discovery perspective, and then from the perspective of the evolving innovative approaches, novel approaches to tackle it. because this has been listed as a major health challenge, and it is one of the existential threats to the humankind. If it is not tackled in well in time, it might very well evolve as a major threat, which can go out of hand, as we have seen in case of the coronavirus pandemic. So this might erupt in another form of pandemic in future, and this might land us in more trouble, as we have witnessed during this pandemic over the past 1 year. So now I'll be sharing my parts, this presentation. Hope my presentation is visible.
Yes, sir, it is visible.
All right. So the topic is countering the rise of superbugs through antimicrobial resistance. A whole lot of new terminologies have been used to define this particular phenomena. Some people say that we might land up in post-antibiotic era if this goes unabated, if this goes unchecked, and we might end up in an antibiotic apocalypse, and wherein there even a small influenza could lead to the death of a person, and wherein there will be no effective antibiotics, antimicrobials available for post-surgical patients, available for patients in intensive care units, available for newborn infants, and that might result into large number of deaths. And that's why people are using several terminologies, like some are using the term as superbugs. These are the bugs, these are the microbes which are resistant to multiple drugs. Pan-resistant, multi-drug resistant bugs or microbes are known as superbugs. For example, we have seen in case of tuberculosis, there are strains which are multi-drug resistant strains. Some strains are extensively resistant strains. Some strains are totally drug resistant strains. And similar types of superbugs might evolve with the passage of time with respect to other diseases, with respect to other microbes. And that might eventually end up in an era where there will be no effective antimicrobials left. Even today, the only last resort that we have are the carbapenems. But many countries have witnessed resistance to even carbapenems. And then we will be left with only one drug, that is colistin. But still the resistance has again been reported against colistin also from several quarters of the world. So the situation is becoming quite alarming and grim in that regard, and therefore needs right solutions, right approaches, innovative solutions to handle it, to tackle it. Now, this is the brief overview of my presentation. First, we will be discussing, I'll be giving you the definition of antimicrobials, then the definition of antimicrobial resistance, what it is all about. We will have a look at the timeline of antibiotic discovery, and then we will look into the mechanisms of antimicrobial resistance. That will be followed by the timeline of antimicrobial resistance. We'll dwell into the causes and modes of spread of antimicrobial resistance, and discuss how, to what extent they are being used and misused and overused in health, agriculture, and veterinary sectors. Then we have heard about the national action plans, the global action plan devised by WHO for containment of resistance, and we'll see what are the strategies which have been propagated at international level, national level, regional, community, and individual level for prevention and control of antimicrobial resistance. And then we will see what are the various novel approaches and innovative methods to combat and tackle antimicrobial resistance. This is the brief overview of my presentation, but I begin with the golden words of the father of antibiotics, Alexander Fleming, who discovered penicillin. In his acceptance speech, he was awarded Nobel Prize for this path-breaking discovery, for this path-breaking invention. And in his Nobel Prize acceptance speech in 1945, Alexander Fleming said, "But I would like to sound one note of warning. The time may come when penicillin can be bought by anyone in the shops. Then there is the danger that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug and make them resistant." Actually, we all know that penicillin was actually synthesized in 1928, but its manufacture at large scale started in 1940s. And the Nobel Prize was awarded to Alexander Fleming in 1945. In his acceptance speech, he had well in advance listed out the dangers, the threats because of the overuse or underuse of the antibiotics. And that is exactly what is happening as of today. These are the things which are unfolding, as a result of which there is antimicrobial resistance. Let me give you another overview of the whole problem and its solution before we go into the things stepwise. Let us have a major overview of what the problem exactly is and what is its solution. As we all know that multiple classes of antibiotics were developed and launched right from 1940s to 1980s, which included beta-lactam antibiotics like penicillins, cephalosporins, carbapenems, aminoglycosides like tobramycin, tetracyclines and macrolides like erythromycin, glycopeptides like vancomycin, polymyxins like colistin, and fluoroquinolones like ciprofloxacin. But since 1990, there hasn't been much progress. Only 3 new novel class antibiotics have been launched. That means over the last 30 years, not many novel antibiotics, novel classes of antibiotics have been launched. We have only 3 classes, that is plurimutilins, lipoglycopeptides, and oxazolidinones. Although many new derivatives of older classes have also been launched, but the need is to launch novel classes which have new mechanisms of action, which have which are new chemical entities. And therefore, though from 1940 to 1990, resistance to new antibiotics took approximately 2 years to develop against beta-lactam classes, but in some cases it took around 16 to 30 years. So for example, in case of penicillin, the resistance was visible within 2-3 years. But in case of other drugs, it took several years. Linezolid also showed resistance very immediately after its launch in 1990. And in 2019, the WHO, World Health Organization, has identified 32 antibiotics which are in clinical development. But out of these 32 new antibiotics in clinical development, only 6 were classified as innovative. This was identified only 2 years back in 2019. And that's why WHO has declared that antimicrobial resistance is one of the top 10 global public health threats facing humanity. Now, the need to overcome this, there is need for innovative vaccine technologies, such as reverse vaccinology, novel adjuvants, rationally designed bacterial outer membrane vesicles, and together with progress in polysaccharide conjugation and antigen design, these have the potential to boost the development of vaccines targeting several classes of multi-drug resistant bacteria. These are some of the new technologies which are being developed, which are being explored to overcome antimicrobial resistance. We'll be talking more about these technologies towards the end of my topic. And then there are new approaches to deliver small molecule antimicrobials, antibacterials into bacteria, such as by hijacking active uptake pathways and potentiator approaches, along with the focus on alternative modalities, such as targeting host factors, blocking bacterial virulence factors, using monoclonal antibodies, using phage technology, using probiotics, using microbiome interventions, and using newer molecules from the natural world, the natural products. All these are also very promising technologies which are being developed and explored, and which are being looked into to see which one is effective. Now, let me take you back into the history of antibiotic and vaccine development. Like till 1980s, we had the conventional vaccine technology. We have seen in coronavirus COVID-19, vaccine was the only effective modality. Rest, nothing else worked. We made use of several drugs like dexamethasone, hydroxychloroquine, remdesivir, and many other drugs. But nothing was very effective unless we got the vaccine. and conventionally vaccines are believed to be the most effective modalities. But till 1980s, we used only conventional methodologies to develop these vaccines, either by killing the microorganism, either by using the live microorganism, or by attenuating them. And it was started way back in 1796 by Edward Jenner, that was followed by Louis Pasteur in 1880, and then by Maurice Hellman in 1960. Maurice Hellman is known as the father of modern vaccines, who has developed more than 40 vaccines. And in 1980s, then it all changed when we had recombinant DNA technology and we had conjugates in place. As a result of conjugates, several new vaccines could be developed, like HIV, like MEN-C, MEN-A, and new genes could be found which could be utilized. Similarly, recombinant DNA technology was used for HBV, HPV, pertussis toxins. OMV, that is these outer membrane vesicles, were used in HIV vaccination. VLPs were used in HBV and HPV. Similarly, viral vectors were used in dengue. RNA and DNA was also used, and structure-based antigen design was also used. So this took a new turn and new twist after 1980s, once these recombinant DNA technologies were in place and conjugate-based technologies were in place. And this continued till 1990s. In 1990s, we had a new set of innovative technologies. For example, bioconjugates, we had reverse vaccinology, adjuvants, structural vaccinology, and synthetic biology, which led to several other vaccines, though some of them which are listed in the outer circle of this diagram are still under development, and some are believed that they may not ever be developed because until there is a huge demand in the market for them. So, but they hold a lot of promise. That is, they can be used to develop vaccines for a whole lot of diseases which are affecting the humankind. And by another few years' time, they could lead us towards the potential solutions to antimicrobial resistance also. Now, coming to the basic definition, so I have given you an overview, a picture of what is the problem and what are the possible solutions. Now, let us go back to the basics, because I do understand that many participants in this particular webinar might be students, scholars who might not be well versed with the basic differences. So antimicrobials, all of us know, include antibiotics, antivirals, antifungals, antiparasitic agents, and medicines used to prevent and treat infections in humans, animals, as well as plants. While as antibiotics is a more specific term, which denotes the chemical compounds that inhibit bacterial multiplication, which are known as bacteriostatics, or which kill the bacterial species, that are known as bactericidal agents, and are mostly natural products synthesized by the bacteria or fungal species as chemical weaponries to kill of other microbes in the nearby macro environment, and to create a balance in the microbial communities in natural ecosystems. So actually, these antibiotics are produced by the microorganisms in order to survive, in order to attract, get the best of nutrients and to keep other microorganisms away. So create an ecosystem which will facilitate their growth and propagation. And the most of these, we know that there are many antibiotics which are semi-synthetic and some are completely synthetic also. Not all antibiotics are natural products, but most of the antibiotics work broadly by inhibiting cell wall synthesis or assembly, by interrupting cell membrane integrity, by preventing DNA, RNA, and protein synthesis, and by interrupting essential cellular metabolic pathways. These are the major mechanisms by which antibiotics work. And this picture shows the major targets of antimicrobial agents in bacterial cells. For example, penicillin, ampicillin, carbanicillin, aztreonam, etc., and cephalosporins, they act on the cell wall synthesis. Polymyxin B, colistin, daptomycin works on it affects the cell membrane structure. Similarly, there is neomycin, chloramphenicol, erythromycin, clindamycin, and lincomycin, which affects the 50S ribosome. While as streptomycin, spectinomycin, kanamycin, gentamicin, and tetracyclines affect the 30s ribosomal fraction. Nalidixic acid, ciprofloxacin, and novobiocin affects DNA gyrase. Rifampin and streptovaricins affect RNA polymerase. Actinomycin affects RNA elongation. And then there are sulfonamides which affect the dihydropyridine synthesis pathway. And the mupirocin and puromycin affects the tRNA. So there are different mechanisms by which these antimicrobial agents work. They have different major targets by which they kill the microorganism. And when we look at the history of antibiotic discovery, we see that there are several ages, there are several eras by which the antibiotics have gone through. For example, there are the dark ages when there were no antibiotics before 1928, the pre-antibiotic era. Then there was the primordial, the advent of chemotherapy via the sulfonamides in 1940s. The golden era, the halcyon years when most of the antibiotics used today were discovered, that is from 1950 onwards. Then there were the lean years in which there was not much progress. And during these lean years, the period is from maybe 1960 to 1990. And then that was followed by the pharmacological period in which attempts were made to understand and improve the use of antibiotics by dosing, administration, etc. The biochemical era was the one in which knowledge of the biochemical actions of antibiotics and resistance mechanisms led to chemical modification studies to avoid resistance. Then there was target era, wherein mode of action and genetic studies led to the efforts of designing new compounds. And then that was followed by the genomic era. High throughput studies were done, and genome sequencing methodology was used to predict essential targets for incorporation into high throughput screening assays. And lastly, there was the disenchantment era. Disenchantment because of the failure of the enormous investment in genome-based methods. Many companies had discontinued their discovery programs because they were repeatedly able to find the same compounds again and again. Now, as I said, till 1990, there was considerable progress, but from 1990 onwards, only 3 novel classes could be identified by WHO. Not much progress has been made. And in this picture, you can see the details about various antimicrobial agents and their period when they were developed. So the first antibiotic or antimicrobial agent was the salvarsan, the arcifenamine, which was developed before 1910. That was followed by penicillin beta-lactam antibiotic in 1928, and then Prontosil, a sulfonamide in 1930. And then many other antimicrobial agents were identified, were synthesized or discovered from the natural world. And till this picture shows you the progress till 2020. And over the last 10 years, that is from 2010 onwards, we have seen the advent of ceftaroline in 2010, fidaxomicin in 2011, bedaquiline, antitubercular drug 2012, delbavancin in 2014, tedizolid in 2014, oritavancin in 2014, ceftazoline and tazobactam in 2014, and delafloxacin in 2017, meropenem and vaborbactam in 2017, plazomicin in 2018, cefiderocil in 2019, omedacillin cycline in 2019, and imipenem, cilastatin, relibactam combination in 2019. These are some of the newly approved antibiotics which are found to be effective against some of these bacteria shown towards the right side. And the problem of antimicrobial resistance is there right since the development of the first antibiotic less than a century ago, because there has been an exponential growth in antimicrobial resistance, and which is disproportionate to the rate at which antibiotics are introduced. So there is a gap. It is a race against time. The rate at which new antibiotics are developed is not commensurate to the rate at which the microbial resistance is occurring. It is, we are lagging behind in this race. And this continued resistance is rapidly eliminating treatment options for the patients and is increasing the cost burden also because of the multi-drug resistant infections. And it has reached now 2.2 billion people 2.2 billion annually, the cost of treating multi-drug resistant infections. And the antibiotic resistance has been found in most bacteria, but several bacteria are particularly problematic and are becoming common among hospital-acquired infections. So we use a pseudonym escape pathogens for these, which have become particularly problematic and are becoming very common in hospital-acquired infections. Escape, E stands for Enterococcus faecium, S stands for Staphylococcus aureus, K stands for Klebsiella pneumoniae, A stands for Acinetobacter baumannii, and P stands for Pseudomonas aeruginosa, while as E stands for Enterobacteriaceae. These are the most problematic microorganisms which are leading to a whole lot of hospital-acquired infections, and that is where the main focus is being laid to evolve antibiotics, antimicrobial agents against these microorganisms. And in this direction, in 2017, WHO has published a list of bacteria where new antibiotics to tackle them are urgently needed, and has grouped these bacteria as per their priority into critical, high, and medium priority bacteria in order to encourage research and development of new antibiotics to tackle them. As you can see in this picture, the priority list of antibiotic resistant bacteria. First is the priority 1, which is critical area, wherein there are no gram-positive bacteria, but these escape, which I just now enumerated to you, escape microorganisms, Acinetobacter baumannii, carbapenem resistant, and Pseudomonas aeruginosa, again carbapenem resistant, Enterobacteriaceae. These are very critical, these microorganisms which deserve immediate attention, topmost priority. Then there is second level of priority, which is high, priority 2. In gram-positive category, we have Enterococcus faecium, vancomycin resistant, and then Staphylococcus aureus, methicillin resistant, and vancomycin intermediate resistant. In gram-negative category, for this high priority category, we have Helicobacter pylori, clarithromycin resistant, Campylobacter, fluoroquinolone resistant, Salmonella species, fluoroquinolone resistant, Neisseria gonorrhea, third generation cephalosporin, and lastly, in case of the medium category, we have the third priority. In gram-positive, we have Streptococcus pneumoniae, and in gram-negative, Haemophilus influenza and Shigella species. So these are the high priority antibiotics, list of antibiotic-resistant bacteria where we need to work. And this picture shows you the global impact of antimicrobial resistance on human health. As I said in the beginning of my talk, at present, around maybe 7 to 8 lakh people are dying per year globally as a result of antimicrobial resistance. 58,000 newborn babies are believed to die in India as a result of drug-resistant infections. And there are 490,000 new cases of multi-drug resistant TB, tuberculosis every year. And there are 874,541 total disability adjusted life years due to antimicrobial resistance in European Union per year. So as Jim O'Neill of UK had said, $100 trillion loss by the year 2050 is predicted, and 10 million deaths by the year 2050 are predicted. Only 30% of developing countries have a national action plan, and there are 7 out of 8 travelers return home with drug-resistant bacteria, which is very, very alarming. And it also has a huge impact on the environmental health. That is 65% increase is there in drug concentration in water, will be there by 2050. And 2 out of every 3 antibiotics have been found in 2/3 of the world rivers. And only 8 large R&D based and generic pharma companies have set limits on antibiotics in their wastewater. So there are 7 different antibiotics which have been found to be polluted, polluting polluting Danube. And therefore, there is urgent need for attention to preserve our environment, to protect human health, and also to protect animal health and plant plant growth, because it is affecting everything. We are living in one world, one health, and one medicine, because we are all interconnected and inextricably we are interdependent on each other. So the 3 major reservoirs of resistance are the humans, the plant, the livestock, and then the environment. So we need to pay equal attention, equal focus on all the three reservoirs, because there are two types of bacterial resistance. One is intrinsic and the other one is acquired. In case of intrinsic resistance, the antibiotic never possessed any activity against the pathogen. The resistance was intrinsic. There was no activity from the very onset. But acquired resistance is the one which is achieved through the transfer of genetic material, what we call as horizontal genetic transfer through transduction or conjugation. And this confers resistance to the microorganism. As I said, this is considered to be a process of evolution wherein the microorganisms transmit some genetic material horizontally through horizontal genetic transfer, and let the other microorganisms also survive and propagate by acquiring that resistance against the antimicrobial agents. And there are some bacterial genes which are carried on the plasmids. Plasmids are small, circular, double-stranded DNA molecules that are distinct from chromosomal DNA. And these plasmids are transferred from one bacterium to another through conjugation. And this exchange can occur between bacteria of different species. Therefore, the strategies that bacteria use to develop acquired resistance are encoded in these plasmids and may be classified into four major mechanisms. One is decreased permeability of the cell wall to antibiotics. Second is modification of enzymes to inactivate antibiotics. Third is drug target site changes. And then fourth is efflux pumps that remove antibiotics from the cell. And drug inactivation through enzymes is the predominant mechanism used by bacteria. And the expression of genes that encode these enzymes may be induced either by particular medications, which is known as facultative, or by they may be constantly expressed. That is, they may be constitutive. Now, in this picture, the mechanisms have been elaborated for your convenience. That is, how the microorganisms acquire a resistance. They may either cause resistance by inactivation through enzymes, through drug inactivating enzymes, or they may cause resistance by alteration of the drug target. The drug target site is altered, or plasmids are used to transfer genetic material, or the inhibition of the drug uptake. There may be modification of the cell wall protein, like the porins, and because of which the intake of the antibiotic will be inhibited, or the antibiotic which has already entered may be thrown out, may be effluxed through the efflux pumps, thereby causing activation of drug efflux pumps. These are some of the mechanisms by which these microbes acquire antimicrobial resistance, and few more mechanisms are showcased in this particular diagram, wherein you can see there are these ribosomes lead to the formation of proteins. There may be ribosomal mutations or modifications, and there may be target site modifications or mutations, and there may be other mechanisms like decreased permeability, etc. And these are the examples of drugs which use these particular mechanisms. For example, imipenem, tetracycline, and polymyxin B causes decreased influx of antibiotic into the cell. Tetracyclines, erythromycins, and quinolones increase the efflux, the throwing out of the drug from the cell. Therefore, similarly, there may be these penicillins, ampicillins, carbapenicillins. They use these enzymatic inactivation by degradation of the beta-lactamase. Then there may be these sulfamethoxazole and trimethoprim, which causes bypass pathways of this tetrahydrofolate. And there may be other mechanisms like phosphorylation, adenylation, acylation, and gluthionylation. For example, by drugs like streptomycin, spectinomycin, kanamycin, gentamicin, and chloramphenicol. And then there may be target site alteration by nalidixic acid, ciprofloxacin, and their target sites are also shown. Similarly, polymyxin B targeting LPS. Then I was telling you that some of the antibiotics, it didn't take them more than 2 to 3 years to acquire resistance, but for some, it took several years. For example, penicillin, shortly after its large-scale manufacture was started, resistance erupted. In case of erythromycin, it took around 13 years. In case of gentamicin, it took around 20 years. In case of vancomycin, it took quite longer, around 30 years. And in case of linezolid, it didn't take even 1 year. But in case of ceftaroline, which was developed in 2010, the resistance developed in 2011 itself. And when we look at the antimicrobial resistance timeline, we observe that there was this eruption of New Delhi beta-lactamase 1 enzyme. There was this strain discovered in 2008 in a Swedish man of Indian origin. The first case of a bacterial infection with resistance caused by NDM1, that is New Delhi metallo-beta-lactamase, a powerful enzyme that gives bacteria resistance to most antibiotics, was discovered in 2008. It grabbed the national headlines in many newspapers, and then it was subsequently detected. The same, this resistance was detected in USA, UK, Japan, and Canada, and many other countries. The last line antibiotic 2 was found to be toxic for human use in 2018, and E. coli containing MCR1 gene was discovered, a gene which is resistant to even colistin, a last line antibiotic that until recently was considered too toxic for human use. But now colistin resistant infections have now been detected in at least 30 countries. And I was telling you that colistin is the last resort if there is carbapenem resistance also. But even in 30 countries, we have seen that colistin resistance has been seen. Then 58,000 newborn deaths in India as a result of mostly pneumonia or some other diseases. By 2015, the WHO, World Health Organization, came up with global action plan on antimicrobial resistance. Prior to that, in India, we had national policy on antibiotics usage in 2011, which was modified in 2017. And then it was evolved into National Action Plan in 2018. And in 2018, there were 33,000 annual deaths from drug-resistant infections. And this superbug gene was found in more than 100 countries, NDM1, which allows the bacteria to evade last resort antibiotic. So by 2030, the economic cost of antimicrobial resistance is believed to be 3.4 trillion, while as by 2050, it is going to escalate to $100 trillion. Now, these are some of the genomic elements, some of the microbial targets, some of the molecular targets of these microbes. As you can see, the midline shows the dates. Below the midline, these are the sensitive microorganisms, and above the midline of showing the dates, the blue line, above that are the resistant, the drugs which have become resistant, the microbes which have become resistant to these drugs. So you can see the sensitive strains were more in 1960 and resistant strains were less. But as we move to 2010, as we move towards right, the sensitive strains have been constantly decreasing and resistant strains have been constantly growing. Now, a whole lot of drugs are resistant and very few drugs are sensitive. So this transformation is there, and there is a detection of a whole lot of genes which are shown on the top, the resistance genes and their genomic elements where they act. So these a lot of work needs to be done in this area, molecular biology. We need to identify more and more such resistance genes so that they can be tackled by appropriate technologies, by appropriate modalities. Now, coming to these action plans, in 2011, we had the first national antibiotic policy that was followed by inter-ministerial consultations on containment of antimicrobial resistance in 2017. And 2015, WHO came up with the Global Action Plan on Antimicrobial Resistance. And following our National Action Plan in 2018, which was devised by the Government of India, Ministry of Health and Family Welfare, Several states also took lead. Kerala was the first to take lead and devise its antimicrobial resistance strategic action plan, which was followed by Madhya Pradesh and then Delhi. And many other states are also in the process of developing their own state action plans for containment of antimicrobial resistance. And there is this guidance document also published by the Ministry of Health and Family Welfare, how to devise state action plans for containment of antimicrobial resistance. This is available online, can be utilized by the states to devise their own action plans. And there is also one document on the monitoring and evaluation of global action plan on antimicrobial resistance, which has been drafted by WHO, by which you can monitor and evaluate the progress in terms of the global action plan in your country. So these are the various official documents available on this. A whole lot of strategies have been devised in these documents in order to contain the antimicrobial resistance. So the state action plans for containment of antimicrobial resistance, the following 6 strategies have been prioritized as per the National Action Plan for Antimicrobial Resistance, wherein it has been propagated and promulgated that there is need to improve awareness and understanding of antimicrobial resistance through effective communication, education, and training. That is the first step. Then there should be strengthening of the knowledge and evidence through surveillance and laboratory strengthening. And then we need to reduce the incidence of infection through effective infection prevention and control measures. We need to optimize the use of antimicrobials in human health, animals, and food. We need to promote investments for antimicrobial resistance activities, research and innovations for containing AMR. We need to strengthen states' commitments and collaborations on antimicrobial resistance. These are the 6 basic priorities of NAP AMR form, which form the basic framework for developing the state action plan. And WHO has also shown us where are the reservoirs and what is the chain of spread, how it spreads along the food chain, how the microbes which have acquired resistance are transmitted from humans to animals, to livestock, to plants, to environment. And then how the overprescribing of antibiotics aggravates the problem. And WHO has emphasized on the need to stop overprescribing of antibiotics, to let the patients finish their treatments, aware them, inform them, educate them, and then how overuse of antibiotics in livestock and fish farming has aggravated the problem. And then there is poor infection control in hospitals and clinics, which needs to be addressed. There is lack of hygiene and poor sanitation, which needs to be overcome. There is lack of new antibiotics, which needs to be developed by the pharmaceutical companies. And if we use these strategies which have been devised by WHO, a lot can be done. Though new modalities can be developed, new approaches can be also adopted, but unless and until we do not streamline the usage, unless and until we do not take preventive measures, control measures, we do not make efforts towards creation of awareness, towards maintaining health and hygiene, sanitation, it will be very difficult to control the spread of antimicrobial resistance. And that's what most of these documents have also emphasized, that there is need to follow up locally developed antibiotic guidelines. We need to have hospital infection control policies, hospital antibiotic policies. We need to have standard treatment guidelines. We need to have in place, we need to have hospital formularies, but unfortunately in developing countries like India, we don't have standard treatment guidelines. It has been established through scientific studies that less than 40% people in public domain are using antibiotics as per the guidelines, and less than 30% antibiotics are prescribed as per standard treatment guidelines in the private sector. It has also been estimated that less than 1/4 of the people are complying to the instructions given in the prescriptions, and less than 1/3 are taking prescription drugs only on prescriptions. So there are these issues. The studies, the systematic reviews, meta-analysis have given very alarming results, wherein these results are showing that there is, particularly in case of India, we don't have sufficient records, sufficient documents, sufficient evidence and data regarding what is the extent of use of these antibiotics in the livestock, in the veterinary sector, in the agriculture sector? What is the extent of use in of these antibiotics over the counter? And then we don't have standard treatment guidelines in many of our hospitals. We don't have antibiotic policies in, but now, of course, the awareness is getting more and more, and people are working in this direction. The leads have been taken by private sector hospitals and many leading government sector hospitals. Hopefully the scenario will improve, the scenario will change in the coming years, but this is the right time when we have to take concrete action, because if we don't curb the misuse and overuse of antibiotics, the sale of antimicrobials over the counter, and if we don't curb the underuse and overuse of antimicrobials by patients, if we don't tell them to comply to the prescriptions now, we will be landing up in a big mess, because India is a huge country, and where if major population resorts to overuse or misuse or underuse of antimicrobials, we will be faced with an enormous tragedy later in years to come, God forbid. And therefore there is need to develop new these antimicrobial drugs. There is need to maintain the balance of microorganisms in human body. There is need to intensify sewage treatment measures, strengthen bacterial resistance surveillance, reduce unnecessary antimicrobial use, controlling environmental pollution, then enhancing community services, and screening and intervening of this MDRO colonization, and multidisciplinary antimicrobial interventions in primary care are also needed. So there are multi-pronged activities, interconnected activities, which need to be taken by different stakeholders at different levels. Unless all the stakeholders join hands and take measures at different levels, it will be very difficult to control antibiotic resistance. Now, ladies and gentlemen, I come over to the novel approaches, the innovative technologies which can be used to overcome and combat antimicrobial resistance. Let me tell you about the Teixobactin, the new antibiotic which was developed, the first member of a new class of antibiotic, and wherein a new technology was used, wherein we resorted back to the dark matter, because there are certain microbes which cannot be cultured inside the laboratory. They have to be taken back to their natural habitat, taken back to the soil. So a device named as iChip was used to culture soil bacteria, allowing the nutrients and signal molecules to pass to the bacteria and then stimulate its growth. The bacteria were sandwiched between two semi-permeable membranes. First, they were diluted so that we have one bacterium in each well, and then these wells were sandwiched between two semi-permeable membranes and placed in the soil, so that these bacteria could propagate, they could grow and develop on their own in the natural habitat. And this is what resulted one of 25 new antibiotic compounds, that is Teixobactin, was discovered using this new method, and was considered the most promising compound, which was toxic to bacteria but not to the cells of the mammals. And it works against gram-positive bacteria such as methicillin-resistant Staphylococcus aureus, but not against gram-negative bacteria such as E. coli. And how does it work? It works by a unique mechanism of action, targeting lipid molecules bacteria use to build their cell walls. And this is how, in the next picture, I'll be showing you in more detail, that first we collect the environmental sample, then we place the iChip plate in a diluted environmental sample to capture only one bacterium per well. And then we enclose this iChip plate in a breathable membrane that allows for bacterial communication. And lastly, we place this iChip device back into the environment, and where small molecules and metabolites can promote bacterial growth. So we are going back to the dark matter. This study was published in Nature, where this is giving rise to promising new antibiotics when we resort to the natural habitat, to the dark matter. And it was also observed that there are new antibiotic resistant genes found in soil microbes in distant places in Norway and in the distant areas where there is no habitation, when there is no population. But the discovery of peptides, enzymes, and other gene products that confer antibiotic resistance has given certain clues to how exactly the antimicrobial resistance develops. Because in areas like the Alps of this Norway, where nobody traverses, the bacteria were found to have developed resistance. and the reason was attributed to the defecation or to the birds, which bring the microbes which are already resistant or the genetic material and then pollute the environs. And so there is hardly any part of world which is safe and secure now from this antimicrobial resistance, even if it is not inhabited by a large population of the humans, but still there may be these genetic products or the material which lead to antimicrobial resistance. Another novel approach is to develop these new antibiotics is from the secretions of toads, Anura and Buffonidae. And there are therapies which are based on bacteriophages known as these are the viruses that invade bacteria and disrupt the metabolism, leading to bacterial lysis, or has also gained a lot of attention. So bacteriophage technology is being developed. This is being looked into. Another option is the research for new chemical entities which are able to inhibit growth and hence overcome the resistance that pathogenic microorganisms display. We are in a process of developing new chemical entities and bioactive compounds which are obtained from understudied natural products, which Dr. Abhishek Mathur was pointing towards, such as toxins from amphibians, insects, marine invertebrates, spiders, and reptiles. These toxins could have a potential, hold a promise for unknown chemical entities which could be used against antimicrobial resistance. Similarly, the aqueous soluble fraction of the parotid gland secretion and the methanolic chicken extract from the Sudan toad Dutaphyrinus melanostitis has antibiotic potential against Bacillus cereus, E. coli, Klebsiella, Salmonella, and many other microorganisms. However, no activity was found against MRSA. So these are the new sources of antimicrobial agents, new leads. We have to resort back to nature. We have to revert back to natural products and amphibians, insects, marine invertebrates, spiders, and reptiles. And then there is research at the molecular biology level, wherein new targets are being explored, are being discovered. So in this picture, you can see these are the various genes, various targets. In the green color are the old targets or activators. In the red color are the new targets, and in the blue color are the new targets for these new antitubercular drugs. So you can see a whole lot of, I'm sorry, I don't have time to expand on this and elaborate more on this. But if you visit this particular paper published in the Frontiers in Cellular Infectious Microbiology, you will be able to get more details. What are the new molecular targets and how best they can be utilized to overcome antimicrobial resistance, particularly in case of tuberculosis. So new molecular targets are being explored, they are being searched, and new bioactive natural products from bacteria are being also searched and identified via a process which is known as bioprospecting. Then there are other methods of genome mining and metabolic engineering. In case of bioprospecting, we explore different leads by subjecting them to a series of bioassays. So we get new leads, new molecules, new micromolecules from natural products. And for this, we make use of bacterial isolates. These isolates are subjected to genomic studies and genomic mining, which leads to dereplication, structure, and activity isolation. And then there are several drug discovery pipelines. Actually, the drug discovery pipeline is running dry. Not many new drugs have been discovered with new novel classes of action over the past 30 years, but still, there is this bioactivity screen obtained from the extracts of bacterial isolates, which can be utilized to find new leads through this bioprospecting and bioremediation, and then genome mining and metabolic engineering. So again, I don't have time to expand on this and elaborate on this, but the paper has been published in Microbial Biotechnology in 2019, where from more details can be obtained. Then this phage therapy is being explored to use phage viruses that may or may not be genetically engineered or phage enzymes in order to destroy the bacteria. And there are also other strategies relying on the probiotics in recruiting, enhancing, or replacing our normal flora with beneficial bacteria to keep disease-causing bacteria in check. And another technology is the CRISPR technology, that is clustered, regularly interspaced, short palindromic repeated DNA sequences, which offers the possibility of using a bacteriophage hidden in a probiotic to cause the bacteria to make lethal cuts to their own DNA. And the benefit of using a bacteriophage capable of targeting and killing a single species of germ would be to leave the good bacteria intact, in contrast to the way broad spectrum antibiotic therapies work by killing both good and bad bacteria. So here, the bacteria will be making lethal cuts to their own DNA, but good bacteria will be left intact. And it will be making use of bacteriophages as well as probiotics. Additionally, innovative solutions such as fecal transplantation have also been explored successfully to cure recurrent C. difficile infections, and immunotherapeutic treatments are also in development. However, most of these strategies are still at an early stage and are not very routinely used. The development of host immune response biomarkers, allowing the diagnosis and measurement of the degree of immune failure, would also be useful to identify patients at increased risk of these healthcare infections in order to implement infection prevention measures. Now, dear participants, the overuse of antibiotics, as I said, is not only by the humans, but also overuse of antibiotics is also in case of important flora, fauna, and ornamental plants. For example, oxytetracyclines and streptomycin are being used for this purpose very rampantly. Streptomycin is sprayed on the surfaces of plants like apples, pears, and ornamental plants for control of the fire blight caused by bacterium Erwinia amylovora. And universally, the market consumption rate of antibiotics is assessed to range from 0.1 to 0.2 million tons yearly. And in 2010 alone, about 63,151 tons of antibiotics were used by livestock alone globally. And these antibiotics are extensively used in medical, veterinary, and agricultural platforms, not only for therapeutic prophylactic, but also for metaphylactic, that is when you are trying to prevent disease in a large population well in advance by giving them for preventive measures, and growth promoting purposes with beta-lactam antibiotics like penicillins and cephalosporins. And this overuse of antibiotics, it is essential to know that hospital is not the only source of antimicrobials present in the environment. The community sewage is a bigger source, and notably, antibiotics are used to increase the protein content and reduce the fat content of meat during breeding. Antibiotics are also used for beekeeping and fruit production. And epidemiological studies have suggested the overconsumption of antibiotics, which further initiates the development of resistance. And when antibiotics are consumed in excess, they remove these drug-sensitive bacteria, and resistant bacteria are left to proliferate. Therefore, antibiotics are overprescribed worldwide, despite the warnings regarding this issued by the World Health Organization. And this extensive antibiotic use in agriculture and veterinary sciences is believed to advance the resistant strains. And then there are certain factors such as drug pharmacokinetics and physiochemical characteristics of the animals, biological processes which influence the occurrence of antibiotic residues in animal products. So the spread of resistance to humans via animal products has existed for more than 35 years, especially when farmers used to harbor similar these agents. Similarly, there was this study by the CSE, Center for the Society for Environment. They used they observed growing antibiotic resistance in humans because of large-scale indiscriminate use of antibiotics in poultry industry within India. 70 chicken samples from Delhi NCR region were tested positive for 6 commonly used antibiotics. 40% samples tested positive. Residues of more than one antibiotic was found in 17% samples. And this pointed to large-scale unregulated use of antibiotics as growth promoters by the poultry industry. And India has no regulation on controlling antibiotic use in the poultry industry. Therefore, India has not any set standards or limits for antibiotic use in chicken. And this, because of this, we'll have to implement a comprehensive set of regulations, including banning of antibiotic use as growth promoters in the poultry industry. So we need to take measures apart from novel approaches to develop new leads, new molecules. We need to take measures at international level, at national level, at community level, as well as at individual patient level. At all the levels, all the stakeholders have to work together, take concrete, concerted measures to overcome this antibiotic resistance, antimicrobial resistance. We need to have rational use of antibiotics. We need to stop over-the-counter use of antibiotics. We need to have standard treatment guidelines for. We need to have antibiotic policies, good standards of hygiene, and then we need to have good hand hygiene practices. We need to use effective diagnostic and treatment modalities for the infections. We need to improve the antimicrobial quality and supply chain. There is a role very well defined under the National Action Plan as well as under the Global Action Plan for physicians, for nurses, for pharmacists, for patients. For everyone, discreetly, there has been very clearly, the role has been defined, what actions they can take. And then there are other measures for pharmaceutical promotion, which is very rampant in countries like India, that we have to curb the unethical practices of promotion, particularly with respect to antibiotics. Now in India, we have this Schedule H1, which is specifically made for antibiotics, antimicrobials. So any unauthorized prescription and use of these drugs is prohibited. Therefore, similarly, we need to restrict antibiotic use in animals. We need to have rapid understanding of the mechanisms of antimicrobial resistance, and then resort to innovation in new drug development and technology. Lastly, the role of the pharmacist. The pharmacists serve as a medication expert on healthcare teams, and given their rapid expansion of their professional role, they are optimally positioned to assess treatment necessity and appropriate antibiotic selection. Their knowledge of antibiotic mechanisms of action and strategies used by resistant bacteria enables pharmacists to make evidence-based decisions about drug therapy, even without formal training in infectious diseases. The Society of Infectious Diseases recognizes that every pharmacist can contribute to the combating of antimicrobial resistance, and therefore objective data should be published which illustrates the impact of pharmacists in these activities, and a literature review that investigated the clinical and economic impact of pharmacist-led interventions related to antimicrobial use has shown a decrease in antibiotic cost up to 95%, as well as reduction in readmission, mortality and infection rates. And selection of the most appropriate agent has positively affected patient outcomes in this study and ensured the preservation of available antimicrobial resources. Therefore, the involvement of trained, qualified, and eligible pharmacists is very much needed. And then only the antimicrobial resistance could be controlled to a large extent. Besides, there is a defined role for the nurses, for the healthcare professionals, for physicians, as well as for common patients. Everybody has to stick to his role in order to overcome this problem. And the qualified pharmacists being the mainstay, because they are the ones who dispense drugs. They can give instructions to the patients. They can counsel the patients on appropriate use of the antibiotics and aware them about the dangers of antimicrobial resistance. And when they do so, they will be contributing very significantly towards curbing this menace of. Now, I don't have time. I think I have already spent 1 hour of my talk. I was given 1 hour, but I will leave you with some of these new novel technologies which you can study in more detail after this lecture. In fact, 1 hour is not at all sufficient to cover this broad area, because there's a huge gamut of issues. There is a huge canvas which it covers. It covers a whole lot of areas, including genomics, genetics, biotechnology, molecular biology, and then epidemiology, infectious diseases, etc., bioinformatics, chemoinformatics. But 1 hour is not at all sufficient. Very soon, there is going to be an international symposium to be organized by the Amrita University for 3 days. So you can have, you can discuss all these issues and you can listen to the experts in more detail. But I will leave you with these newer technologies, novel approaches, which you can study at your own level. One is recombinant DNA technology. Then there is glycoconjugation. And the third one is the reverse vaccinology, then structural vaccinology. And there is bioconjugates and genetically modified, there's outer membrane vesicles. And lastly, we are all living in one world. Therefore, there is this idea of one medicine and one health propagating, where this one health is the umbrella which covers everything from public health to veterinary science to plant science to botany to zoology to ecology and health economics. And this idea was propagated for the first time by Rudolf Virchow in 19th century, the German physician and pathologist. And this was further propagated and further taken ahead by Sir William Osler. Because this is an interesting fact that Sir William Osler used to teach medical students at McGill College, and he used to teach veterinary students at the Montreal Veterinary College. So we are living in an interdisciplinary world where everything is connected. Social sciences is connected with sciences. That is in turn connected with humanities. We all need to work together in a collaborative, multisectoral, and transdisciplinary fashion in order to work at local, regional, national, and global levels, and to achieve optimal health and well-being outcome, recognizing the interconnections between people, animals, plants, and their shared environment. This is what the idea of one medicine and one health is all about. So this is the international symposium, which is starting from February 24th at Amrita University. The registration is free of cost. And on the left, you can see the issues which they are likely to discuss: diagnostics of AMR, alternative therapeutics, novel strategies to counter AMR, antibiotic stewardship and community health, the mechanisms of AMR, antibiotic usage in agriculture, drug and vaccine development, microbiology of resistance, escape pathogens, drivers of AMR, environment and AMR, microbiome and AMR, molecular studies, nosocomial infections, biofilms, and editing tools in combating resistance. So I'm sorry, I don't think I could do justice with this topic, because I couldn't cover even half of these topics which have been listed. But of course, in a symposium for 3 days, they will be discussed in much more detail by the eminent experts in the field. So if you could join them. Lastly, I have used more than around 50 these references have been used in the preparation of this presentation. I couldn't list them, some of them alongside the slides. So I am showing them towards the end. I'm showing only some which have been prominently used. Thank you very much for patiently listening to me, for your audience. And back to you. I'll be happy to take if there are any questions. Over to you, Swati ma'am.
Thank you so much, sir. Sir, could you please stop sharing the screen?
Yes, of course.
Thank you so much, sir.
Thank you.
Well, I'm feeling really fortunate to have got a chance to get so deep understanding on such an important topic today. Thank you so much, sir, to have enlightened and in this area with your vast knowledge and expertise. Your remarkable depth of understanding of the subject really made the webinar an exceptionally stimulating and informative. It was incredible how you tailored your message for the audience and captivated them from the beginning of the presentation till the end. Thank you again, sir.
Thank you, most welcome.
As I can see here, the audience has really appreciated the session today, and their deep interest has aroused certain queries too. So I request you to kindly address the queries as I read out them for you.
Yes, please. Sure.
Before I begin with the queries round, I request our viewers to kindly spare a few minutes in filling the feedback link that has been sent on the live chat. So the question is that in the COVID treatment, since various antimicrobials have been given to patients as a possible remedy showing therapeutic response against COVID, can there be a possibility that during such treatments, patients could have developed AMR? Could there be a possible solution to this, sir?
Yes, there is a possibility that if antimicrobial agents have been ruthlessly used without following proper protocols, without following standard treatment guidelines, and during the COVID-19, they were the most stressful times, most frustrating times, and anything and everything that was found to be effective was used by the people. starting from hydroxychloroquine, which was eventually found to be of not much use, from remdesivir and favipiravir and remdesivir, dexamethasone, and many other things. And even antimicrobial agents were also used to a very great extent. And in this process, if the antimicrobial agents have been misused or overused in certain societies, in certain regions for a longer period, definitely it would have aggravated the problem of resistance. But there is no way, once the resistance is there, it is very hard to overcome. Once a strain has become resistant in a particular person, it is very hard to overcome that, unless, as we were discussing, you have certain novel and innovative technologies in place. But I hope that, since the enlightening came in the beginning that it is a virus, so it may not be effective against the antibiotics, may not be useful. So that much awareness was there from the very beginning. Therefore, people did not use many antibiotics because they knew it's a virus which may not be showing activity against antibacterial agents. But maybe there were some concomitant infections, there were some comorbidities, as a result of which many people might have used antimicrobials, but there is nothing which we can do if it has led to any antimicrobial resistance in those populations.
Thank you very much, sir, to have answered the query. I request Dr. Mathur to kindly ask if there are any queries from his side too.
Yes, please.
Thank you, ma'am. It was a great lecture, sir. First of all, congratulations to you and thanking you for being here and letting us from the great information you described. There's one question: can prescribing polypharmacy, which includes 2-degree antibiotics, helps in bypassing antimicrobial resistance?
Polypharmacy, no, polypharmacy itself is a problem. Polypharmacy is a condition in which there are multiple drugs are prescribed simultaneously. More than 7 to 8 drugs are prescribed simultaneously, most of which may not be actually required most of the times. So polypharmacy is a menace, is a problem in itself. But if it is if it can only add to antimicrobial resistance, because polypharmacy may also include irrational use of antimicrobials, antibiotics, and that may we have seen in our settings in J&K, we have non-qualified people manning the pharmacies who do not have much knowledge about antimicrobial agents because they are not sufficiently trained about it. And whoever goes to their shops, they immediately dispense antimicrobials, antibiotics, whether they are needed or not, without any expert consultation. And that too, the antimicrobials of high order, for example, even they do not hesitate to use carbapenems, which are the last resort, the mainstay of bacterial resistance. If God forbid they also become resistant, then we will be left with no solutions. And then there will be deaths in ICUs, deaths in post-surgical wards, deaths among newborn infants. And it will be very hard to treat. Even a small influenza can lead to serious morbidity and mortality. But polypharmacy is a problem in itself. I don't think it can in any manner contribute towards reducing antimicrobial resistance.
Okay, sir. Thank you so much. Over to Swati, ma'am.
Yes,
Thank you so much, sir. Thank you so much, sir. I'm really glad you reserved a few minutes to field the question, sir.
No, I can, if there are any more questions, I'll be happy to take any more questions in the chat box if there are.
Sir, the webinar was so self-explanatory and it was so good that it wasn't left with any window to field the questions anymore. So the answers were very insightful and I really appreciate your fervor and patience with which the questions were being dealt. And thank you to all our viewers to have stayed over so patiently and to have enthusiastically participated in this session today. We are gratified by your presence. Thank you so much.
I'm highly thankful to Dr. Shashi Alok ji, yourself, Abhishek ji, for accompanying me throughout this session and for facilitating this lecture, for inviting me. It's a matter of great pleasure and honor. And I would request the participants who have any questions, please mail me, inbox me. I am available on Facebook, on social media. My email ID is there. You can search me, Google me out. And you can please feel free to ask me any number of questions that you have. Wherever I could be of any help, I'll be glad to answer any questions which the participants might have. Thanks again to all of you, and wish you all the very best. SPSR is doing a fabulous job. Every Sunday you are informing and educating people all across the country, and you are spreading knowledge, you are enhancing their capabilities, their skills, their competencies. You are building capacities of our pharmacists. So it's you deserve all kudos and salutations, and you are doing a tremendous job, highly commendable job. Many congratulations, and my heartfelt gratitude to all of you, Dr. Sheshiji. Thank you very much.
Thank you so much. Thank you so much, sir.
Namaskar.
Namaskar, sir. Thank you, sir.